Hello, and thanks to the organizers for inviting me to give this talk. So my work at the University of California, San Diego has concerned extracting information from high-density EEG recording, MEG recording, ECOG recording, human electrophysiology, and behavior. But anyone working in this field, I think, should be continuously connected with the question, what is EEG? We know it's brain electrical activity, but really it's only a small portion of cortical brain electrical activity, and an even smaller portion of total brain electrical activity. We know those things, but what particular portion or portions is it? And how are these portions triggered and modulated? And what is their functional significance? So EEG was first demonstrated in the 20s. By 1937 here, it had come to Harvard. I don't know what kind of experiment that was. 20 years later, Gray Walter showed the first live, real-time analog scalp map. These tubes flickered with the alpha rhythm. 20 years after that, with the advent of computers in labs, the averaging of responses to similar or considered equivalent stimuli gave rise to the whole movement to study ERPs, event-related potentials, which dominated academic EEG studies for the next 20 years. However, by the next 20 years, the continuing advances in computer power, available computer power, and packages such as MATLAB made it possible to image the current distribution over the scalp at one time point, or to make movies of it, to do lots of signal processing with the EEG, and that's when we began putting out functions in the form of MATLAB functions that people could use to on their data, and we called it EEG Lab. 20 years further, and I believe the prospect is in sight. In fact, it's available to perform high-resolution imaging of the effective sources of EEG, both their locations and their time courses in moment to moment, second to second. So, some thoughts about EEG. First of all, our consciousness, including our embodied agency, our feeling of being an agent in the world, a willful agent in the world, makes it necessary that our EEG be complex, because our world is complex. And I wrote a little poem. Brain processes have evolved and function to optimize the outcomes of the behavior the brain organizes in response to perceived challenges and opportunities. Brains meet the challenge of the moment, every moment. In our brains, there's continual cycles of perception, action, and evaluation of the results of our action. This cycling is going on continuously through network activity, supported by network activity in our brain and in our cortex as well. This makes EEG signal processing a real challenge to study how the brain supports our natural cognition as embodied agents in the world. Second, brain dynamics are inherently multi-scale, and I'm thinking about electrophysiological dynamics or electromagnetic dynamics. At each spatial recording scale, the important thing is that the signal that you record on a channel is actually produced by active partial coherence of distributed activities at the next smaller spatial scale. So each single channel is not a channel, it's not the channel, it's a sum of activities at a next smaller scale. So if you were able to record from the bouton of a neuron, you would record potential fluctuations that actually arose through molecular events, synaptic molecular events, and their coherence in time and space. If you record from within the cell, the voltage time series you're recording actually reflects the activity on thousands of synaptic and also non-synaptic sites on the cell surface, as well as the intracellular dynamics. I meant intercellular dynamics. If you record extracellular fields, you're recording the synchrony in time and space of activity in the neighborhood of the recording electrodes. If you record farther away on the cortical surface or very far away on the EEG surface, again you're recording fluctuations in the synchrony in time and space of projecting cortical field potentials. Third, the information flow is bidirectional, from small to big, from big to small. It was thought by the single-cell neurophysiologists who saw the brain working as a telegraph code of spikes put out by neurons, that something like the EEG or the local field potentials were just what they called the roar of the crowd and meant almost nothing. Well, now it's been again shown that local field potentials influence the exact timing of spikes and the exact timing, the synchrony of spikes from coming up from a local area. That means that common targets of those spikes will get a much more effective or less effective signal depending on the degree of synchrony of the outgoing spikes. That will change the import of the message, the effects of the message at common sites, common target sites, and will change learning and plasticity. Therefore, these macroscopic field potential dynamics actually change within neuron receptivity, plasticity, and therefore brain's learning and plasticity. Well, when we come to the EEG, there's been talk for decades about the source of the EEG. Well, the source of the EEG is primarily the cortex because of its favorable orientation and location with respect to the scalp. But what do you mean by a source? It's important to have that in mind clearly. Number 4, the generation and modulation of cortical EEG and ECOG is really a complex topic, and it hasn't been very well studied, and it's not very well understood. It's known, that the relationship between the excitatory pyramidal cells and the inhibitory cell networks within the neuropil are very important for sustaining oscillations. It's known that what Edelman called the value system modulations, these brainstem-centered neurotransmitter-labeled systems that give some import, some news about the import of events, the dopamine, serotonin, noradrenaline, and so on. They're known to modulate the local field potentials within cortex. Neuroglial interactions are becoming understood to be much more important than ever thought before. Cortical connections, other than local nearest neighbor connections, are surprisingly sparse in the brain, but each cortical area is connected to perhaps a couple of other cortical areas, and those play a role in tuning the mesoscale dynamics of cortical field potentials. The cortical thalamic loops also are thought and understood to support oscillatory behavior. And finally, the ephaptic modulations, that is, the effects of field potentials on the timing of spikes, form a feedback loop that can further influence brain network activity at the field potential level. And then finally, of course, on the right we see here the layer structure of cortical field potentials that project to the far field as far away as the scalp electrodes. And they originate in voltage differential between specific layers of cortex. All these facts and their influence on local field potentials was not only not studied, but it was extremely difficult to get funding for any kind of study or even receive tenure for studying it for decades after the development of single-cell microelectrode recordings, as was explained to me by one of the founders of the UCSD Neuroscience. So that it's never really been deeply studied or appreciated. Now here's a time-lapse picture, of course, of the sun at some wavelength, showing the complex electromagnetic activity of the plasma in the sun. And we see features at different spatial scales. If we zoom in, for instance, this picture, which was taken surprisingly by a home telescope, we see that structure is at all spatial scales. We see large features like this sunspot. We see smaller features, and within those smaller still features, and each voxel here is 120 square kilometers. So if we could zoom in still further, I'm sure we would see further fine detail. Well, has such a picture been taken of the voltage activity on the cortex? No. Brain spatial temporal dynamics have not been imaged at high resolution, that is, simultaneously on multiple spatial scales, coarse and fine and very fine. That's something that various brain projects now are working toward. But in fact, it hasn't been observed nor modeled sufficiently. Now, unfortunately, a naive interpretation of the EEG is that the field potential must come straight up from the brain out to the electrodes, and each electrode being sensitive to the area directly under that electrode. Now that, of course, is, almost nonsensically wrong, or only very vaguely a part of the truth. The basic truth, according to biophysics, is shown in cartoon on the right, and so I asked my colleague Zeynep Akel-Nichar to take one of her electrical head models based on an MR image of an actual subject and synthesize a small patch of cortex synchronously producing in this case, a blue negative potential surface on its surface. And then to visualize the projection to the scalp of that effective source. Now, at the distance of the scalp, the signal received in the scalp will resemble almost perfectly an equivalent dipole. That means an equivalent a tiny oriented battery oriented perpendicular to the cortical patch, that whose projection to the scalp is equivalent to that of the patch itself. And the projection you see is to the whole scalp. Every portion of cortex projects to the whole scalp, except perhaps some sort of equator line where the two where the positive and negative poles divide, so that along that line there would be no projection from that particular source. So this is a very broad point spread function. From a point on the cortex, the potential spreads to the entire scalp surface. That's basic biophysics. Now, if one of your channel is always at least one channel, one electrode referred to another electrode. That means the voltage of one channel minus the voltage at the other channel. The difference between those voltages. If your two electrodes happen to be here, then of course this channel would record nothing from this source, because the voltage produced by that source is the same on both electrodes. If, however, this was your channel, well, it would be blue, that's negative, minus green, that's 0. So negative minus 0 is negative. So you'd get a negative potential from this source as it's configured. But if your electrode pair was like this, you were recording from the right side of the head, referred to, say, the vertex, then it would be a 0 potential minus a negative potential. Therefore, it would record a positive potential. So this channel would record a positive contribution from this source, or this effective source. And if the electrodes happen to be here, then it would be, of course, positive minus a negative, which would be still more positive. So you record a larger positive voltage. So each EEG channel is a spatial filter for some sources and not other sources. Well, which sources? So I asked Zeynep to show us that too. So here, for instance, is a channel. You might call it, what would you call it, F4 or 6 to A2 in the language, standard EEG language. And this, again, in a realistic head model taken from an MR image. Let's imagine that you have channel A, electrode A referred to electrode B, or A minus B is your channel. Then it's not true that this channel is only sensitive to cortical tissue directly below electrode A and not electrode B. That's simply not true. Actually, recording the difference in potentials, all the potentials, the sum of all potentials arriving at A minus the sum of all potentials arriving at B. Well, what cortical territories produce such potentials? And here's the answer. Everything in this picture that's not bright green, every piece of cortex that's not bright green in this picture projects voltage to channel AB. Some territories project with the positive sign, that is, whatever the voltage is in the red areas shows up as, contributes positively to the voltage in channel AB. In the blue areas, any potential is there is actually contributes negative of that to channel AB. So the equation is at time t, and given this is the projection map of for channel AB, and given a map of voltages at every voxel, every cortical surface element at time t, then the one voltage value that channel AB records is then the sum over all the volume voxel elements in the cortex of whatever the radial projection is, local field potential projection is from that voxel times the value of that voxel in this projection field map. Now, and that gives you the single value at time t. Well, at a time t plus 1 or t plus n, then of course this projection map won't change, but the VE, the voltages at every element, may change in very complex ways. And again, you perform this calculation and you come down to one value. So this channel, AB, is recording from not one place in cortex, not one network in cortex, but a motley collection of suitably oriented cortical territory. Okay, so that's the reputation of EEG as a very fuzzy brain imaging modality, and the de facto treatment of it as a scalp imaging modality by many of its practitioners for many, many years. Now, what's the idea of an effective source? This is a picture of a pond ripple, and this is Walter Freeman. He put little grids of electrodes on animal smooth cortex about the size of my little fingernail, and recorded their phase portrait, the phase of activity at, he liked to look at gamma frequencies. And he found it usually to be chaotic, but at key moments in the olfactory bulb, when the animal sniffed a new odor, for instance, the phase would reorganize, and you'd get this rippling in or rippling out, which could be rhythmic. The phase portrait changed, and you had this smooth phase portrait where phase was smoothly and slowly varying across the cortical patch. He called them phase cones. Plenz later looked at avalanche events, sort of single events where potential spread through the cortex, and has shown that they occur in brains of animals, and claims in humans as well. Here's a study on Townsend 2015 where they used, I think it was anesthetized rats. So the rats only showed very low frequency EEG because of the heavy anesthesia. But they again put a grid on the cortex, and they looked at the spatial temporal dynamics of the local field potential. And sure enough, they again saw these dynamic patterns, this rippling out, or sometimes rippling in, or other patterns, whirls and saddle bifurcations and so on, and showed that they had systematic relationship also to spiking activity. So, in what sense are these local phase coherence, these islands of local phase coherence, these pond ripple-like occurrences in cortex, effective sources of EEG? So again, I asked Zeynep to make a simulation. I asked her to divide cortex, their cortical surface model, into 1 million independent elements, about the size of a mini column, 100 microns or so in diameter. So you think of it as 100 field potential in 1 million mini columns of cortex in simulation. And I asked her to put the same EEG-like waveforms into all of them, the same power spectrum and the same amplitude, but to make them all phase independent of each other. So they're all producing field potential, 1 over F field potential, but they're doing it independent of each other. And then I asked her to project, and then I asked her to see what the EEG projection would be from that, and then to make emergent coherence, phase coherence, in one small patch of cortex. So not changing the amplitude or the power spectrum of all these million sources in the simulation at all during the simulation, but just changing their degree of synchrony within one small patch. And then this will show the scalp projection, and this will show the change in coherence in that patch from 0 to 1. So here we go, and you see without coherence there's very little EEG projected. Why? Because there's phase cancellation. But when one patch becomes phase coherent, then the net projection to the scalp becomes large. At all times during this animation, all million cortical voxels are equally active. So then why the large change in the scalp EEG? It's because the spatially coherent activity acts as an effective source of the EEG. The actual sources are 1 million, but the ganged phase coherent activity in the small patch acts as an powerful effective source of EEG. So my claim then, or my hypothesis, is that the effective sources of scalp EEG are mainly emergent islands of local synchrony or near synchrony in cortex. Now, why should effective sources of EEG or MEG equally be compact? Well, here I'm going to just quickly go through some physiological plausibility argument. Let's say you had domains in which local synchrony arises in some situation. The excitatory and inhibitory cell networks within this cortical area are highly coupled, and that gives rise to cyclic behavior, both narrowband and broadband. But the inhibitory cell networks have no long-range processes, and that means if this cyclic behavior that they jointly produce is to extend, it should expand neighbor to neighbor, like that. And this is very much Freeman's phase cones or lens avalanche events. The coupling of cortex from between areas, distances larger than a millimeter or a tenth of a millimeter even, is vastly sparser than local connections. So that means that the areas of cortex, by and large, separate, non-adjacent areas of cortex, they don't have any direct coupling that would allow them to be synchronous. Now that's not quite true. Of course, they have common inputs in some cases. If you you can imagine, big events in the brain could produce, some global activity. Although global seizures are not, first of all, not so global usually, and second, of course, exceptional and pathological. In general, brain area, local field potential arising in disjoint areas are not coupled. Now that's not true, of course, and furthermore, the thalamic cortical loops, which is also important, known to be important in sustaining oscillatory behavior, are primarily thin and vertical, radial. These two facts alone, these two physiological facts alone, mean that domains of local synchrony, if and when they arise in cortex, should mainly have independent, have no phase coupling between them. And therefore their voltage projections should be largely temporally independent of each other. And of course, another biophysical fact is that the projection from a compact area of cortex in the brain will have a projection to the scalp, whose spatial pattern resembles exactly that of an equivalent dipole, a tiny oriented battery placed somewhere near the center of the patch. Now, of course, some cortical areas, some cortical patches are densely connected by white matter tracks. And here one can imagine that you might get resonant behavior in which a single waveform was produced jointly by two areas that were bidirectionally coupled by dense white matter. So my own goal closest to my heart in my years of research has been to make effective source, to resolve effective sources, and to make source-resolved high-resolution imaging and modeling of the labile, mesoscale, and network scale electromagnetic brain dynamics, particularly of cortex, from EEG data. And then to use that to study all three aspects of human consciousness. Now, what are the three aspects of human consciousness? And I was cued to this by reading in a book by Meher Baba, The Discourses, where he casually remarked that human consciousness has three aspects, and he called them knowing, feeling, and willing, without further explanation. But it occurred to me that if by knowing we take into account all of perception, all of memory, and attention-related processes, intellectual processing of facts and figures, all of that, and put that in the world of knowing, if we take all the interoception and all the feeling of emotion and all the undefinable aesthetic and religious, spiritual feeling, all the world of feeling that's understood now to be so important in determining our behavior, actually, in determining our motivation and therefore our behavior. If you call that the world of feeling, or consciousness of feeling, then that leaves the feeling of the knowledge of ourself, the consciousness of ourself as an embodied agent in the world, able to affect change in the world through our actions, through our bodily motions, and through our intentions to act. So for too long in the 20th century, experimental psychology was focused on this knowing aspect, learning, memory, attention. Studies of the feeling aspect of the mind are just beginning to come up, and the fMRI opportunity has accelerated that. I'll show here, I think, strong evidence that EEG is also suitable for studying feeling consciousness. And in an effort we've made over the last decade, it's also very useful and usable for studying the willing aspect of human consciousness. Okay, so now we talk about information-based signal processing, and this was a topic that it came to me, and I was fortunate to be involved in 25 years ago now. The question was, if you have a cocktail party, and at the cocktail party, there are sound sources, of course, in the room, and there are some So here's a saxophone and a man talking or singing. And here are some microphones, and each of them, of course, is recording cocktail party noise. Is it possible, the signal processors asked about 30 years ago, to separate out the sound, and each one is mixing, each microphone is recording is mixing the sounds from these, summing the sounds from one of these different sources. Is it possible to separate out from their mixtures the original sound sources without knowing what the sound sources are? So that was called blind source separation, blind to what they are. In other words, instead of trying to find a saxophone in those recordings and a man's voice, just simply find what were the sources of information in the data. If you perform the unmixing so that the waveforms assigned to the two putative sources became more and more temporally independent, with giving separate information to the mixture, then of course you could only, in this case, arrive at the two original sound sources. I read that paper in preprint the paper of Tony Bell and Terry Sinofsky, one late night, and the light bulb came on. I thought, well, if you have some sort of effective source in cortex, or multiple sources, and their potentials sum just like acoustic pressure does at the microphone, so they sum the voltages at the electrodes, then perhaps ICA applied to the EEG would find the time courses of the effective sources and their scalp projection patterns as well. And so 5 hours later, we got together in the boardroom of Terry's lab and made a plan to put out a paper in a week, which we were able to do. And I'm happy to say that the preliminary conclusions we drew from the first look at an ICA decomposition have held up all these 20 years now. And basically the claim was that ICA decomposition of multi-channel EEG data can find distinct EEG effective source activities with relatively high signal-to-noise ratio, and find each of their separate scalp projection maps or patterns. So one thing we saw right away is that ICA decomposition can separate non-brain effective source processes. Well, what are these? Well, we know that, those of us who do EEG know very well that you have eye blinks that contribute large potentials to frontal channels. You have saccades, lateral saccades, for instance, will also contribute largely. Cardiac rhythm can come into the EEG, and then various muscle activities from each neck and scalp muscle will contribute in their EMG activity to the electrodes near to them. So, but now consider this. So ICA does separate these under favorable decomposition conditions, of course. ICA can separate these into and separate them out from the rest of the source data. Now consider, though, that why are these temporally independent? Why can you predict none of the waveforms from any of the other waveforms? Well, because they're functionally independent. What causes the EMG to take that form has nothing functionally to do with what causes eye blink to occur there, or the heartbeat to put potentials in across the head. So temporally independent processes, effective source processes, both non-brain and brain, also tend to arise to do their own thing. That means to have their own function also. So that the independent source processes are functionally independent, and that's what makes them so powerful for cognitive neuroscience, as well as their localizability being useful for neuroimaging. So then we showed, some years later, a beautiful result, that if you take some high-density EEG recordings and you decompose them with some sort of blind source separation algorithm, and we use some 20 of them. The result is that the temporal independence of the component waveforms produced by an algorithm, such an algorithm, is proportional to the degree with which that algorithm, in transforming from channels to component space, removes the mutual information that's in the channel signals. Channel signals where electrodes are neighboring are, of course, highly overlapping, highly correlated. But if you remove all the the more mutual information you remove, the more components you will find that have spatial dipolarity. What does that mean? Here are some components, and what it means is, if a component's dipolar, that means its scalp projection, learned from the data by ICA, without knowledge of where the electrodes are, or anything about electrophysics. Its scalp projection map almost perfectly resembles that of a single equivalent dipole in an electrical head model. Here with only 1% residual variance in that head model. Here with 2%, here with 4%, here with 8%. Here 16%, although this may be, that value may be artificially inflated a little bit by the sparsity of channels on the side of the head. Here 32%, where it becomes impossible for this to be the projection of a single cortical area. Or 64 again, or we had to go to PCA to find this this component projection map. That's impossible to be the projection of a single cortical area. So the more and this is what we found. If you count the number of dipolar, almost perfectly dipolar component maps that you obtained by a decomposition of some dozen 70-channel EEG data sets, that number is almost perfectly linearly related to how much mutual information in the channel data is reduced, is squeezed out, so to speak, of the data as the linear transformation to component space occurs. So, and here's that result. That result cannot be predicted mathematically because it has to do with time and space, which are not in the algorithm knows nothing about space or about time for that matter. It's pure statistics. So this has to do with the origin of EEG and the tendency for the effective sources of EEG to be functionally independent of each other, or near independent of each other. And we see here PCA being the least, producing the least dipolar localizable sources, and Infomax by Tony Bell, extended Infomax, and then Amica by Jason Palman in our lab being the most effective. Now, I showed that these effective sources include these non-brain processes, functionally distinct non-brain processes. But a typical decomposition of high-dimensional EEG data will also have some dozen or more components, processes, whose scalp projections almost perfectly resemble that of an equivalent dipole, which can be the equivalent of a synchronous activity within a cortical patch. Of course, sometimes the independent component maps, or the IC maps, can be exactly modeled by the projection of two equivalent dipoles. In this case, located bilaterally symmetrically, and therefore plausibly connected by perhaps corpus callosum or common drive mechanisms. Now, here is in practice what what IC decomposition under favorable circumstances can look like. Here are 9 out of 100 channels in this data set, a half-hour data set of the subject was performing the a two-back task with feedback. And here are 9 of the 100 independent components, activities, and scalp projection maps in that same 15 seconds that we selected out of the data. So what do we see here? We see a bilateral occipital source, lateral occipital cortex, in which after every trial, an alpha burst occurred. This area was producing alpha bursts. And we can look in the different trial and see trial-to-trial differences. We clearly have high resolution here of trial-to-trial differences. So we can see context-related differences in brain dynamics in individual areas, and we can also look at network effects. Here, for illustration, I computed the ERP for this cortical area just by averaging all 1,000 epochs in the experiment, and then I repeated the ERP for every epoch. So reducing to an average, of course, throws away all the variability, including the phase variability of the alpha collapses. And you see the information collapse by throwing away most of the data in the averaging procedure. But simultaneously, ICA finds, this was AMICA decomposition, finds a frontal midline theta, which was showing theta activity during period of correct performance in this case. Here is a central occipital alpha, which showed a quite different time course of activity from the lateral occipital. Here is a postauricular muscle with EMG and then muscle spikes. Here was a subspace of components that accounted for the upward motion of the eyes during slow eye blinks. The subject was blinking after every trial and then producing this alpha burst. And this was captured in 2 components of the EEG, or an IC subspace. So Jason Palmer showed us, and this had been reported before also, that what ICA is really doing is separating the data, the sources, the effective sources, into subspaces that are within each subspace. There's some residual dependence, or some dependence, but between these subspaces there's independence. Now, if these subspaces are one-dimensional, a single area, static area, spatially static area, then of course we call those independent components. But when we computed pairwise mutual information between the IC time courses, we found that there were subspaces in which mutual information remained. So, for instance, the oval shows 4 muscles that had whose time courses across the half hour had some dependence. Now, mutual information doesn't tell us what kind of dependence they had. And in fact, this whole area remains ripe for investigation by anyone who feels capable of investigating. There's a lot of richness to be studied here. So there has been a lot of talk about low resolution EEG source imaging. How possible is it to perform high resolution EEG source imaging of effective sources? This is from a paper in 2016 by Zainepikale and Achar. And in it we show that an algorithm that allows us to estimate for each individual subject the most important missing parameter in forward electrical EEG head models. And that parameter is the conductivity or resistivity of the skull to current. It's a parameter that is different in each person. It differs across ages, likely genders, but within adults it can vary by a factor of 3, we believe. So in this scale algorithm, we use a source localization technique called SCS for sparse, compact, and smooth. It tries to find sparse, compact, smooth distributed locations for a dozen or more effective sources found in data by for the scalp maps of the scalp maps of a dozen or more effective sources found in the data by ICE decomposition. Here in this, and it estimate the skull conductivity and to put that into the head model to allow more accurate source localization to the cortical surface. So here is just one of the dozen or so component processes, the scalp maps that we used. And we began 2 runs with quite different estimates of how much more resistive the skull is for this subject than the brain tissue. And we used 2 values that are common in the literature, 80 and 25. Scale is a iterative algorithm which at each stage estimates the source localizations using SCS, measures their compactness, and then finds the gradient direction and degree to which change in the conductivity estimate for the skull will make the sources more compact. And following the gradient down, both runs here converge to the same result. And this shows the localization, the final localization found for this component process. And the brain to skull conductivity ratio, which for this subject was estimated at 34. It was that 34 that allowed this component estimate to become compact and better fitting the actual map found by ICE. Here's another subject whose EEG data from some 256 channels was recorded during an hour of them playing a complex multi-screen, multi-touch screen, multiplayer virtual world video game. These are a dozen or more independent component source maps, each compatible with a dipolar representation. And here we're trying to find the actual cortical, to estimate the actual cortical locations of the locally synchronous activity. When one goes, as I did here, to the fMRI atlases online and finds the cognitive processes that these particular source localizations have been associated with in previous fMRI studies, one finds, anecdotally here, things such as multitasking, memory for location, situational responding, spatial interaction, manual agency, visual reasoning and object perception, spatial goal awareness, etc. All cognitive capacities that are exercised fully in this difficult and highly involving video game. Now one might say, well, in an hour of EEG by a person who's very active mentally and physically, you would expect that you might have effective sources in more of the brain. So yes, instead of learning one ICA model for the whole hour, we can set several models as the goal and have them fight over portions of the data to represent. And when you do that using multi-model amica, then you find many more areas that represented in source areas represented in the data. So although this is an anecdotal observation here, it certainly is not a systematic evidence. It is consistent with now some 20 years of results in various cognitive experiments in which the results of ICA, the brain compatible components found by ICA, invariably turn out to be localizable to areas that should be naturally involved in the subject's task, according to all available evidence from fMRI and neuropsychology, etc. Okay, so using information-based signal processing and imaging, we can study cortical field dynamics, supporting human consciousness. But what about human feeling and human willing? Not only human perception, memory, strategy, and so on. Well, I'd like to present again this wonderful experiment that I did with Julie Anton some dozen years ago now, which used emotion imagination. We wanted to understand the EEG dynamics supporting emotional experience. And to do that, we set subjects in a comfortable armchair, had them keep their eyes closed, and listen to it and follow instructions given by audio tape. And the instructions suggested in sequence the imaginative experience of 15 different emotions. We used the method of guided imagery, which I learned from the psychologist Helen Bonney. After an initial relaxation instruction, the subject heard alternating suggestions to imagine scenes engendering the named emotion, and we alternated positive and negative emotions. So for anger, we suggested that they either remember a situation in which they're very angry, or they imagine a situation in which they would be angry. For instance, walking out on the street and finding someone bashing your car windows with a baseball or cricket bat for no reason. And to try to feel that emotion in your body, that somatic feedback loop, that interoceptive ability to association between emotions and feeling and bodily feelings that has been described. That helped the subjects retain the feeling while continuing to hold the imagined scene in mind. When they felt the feeling wane, they were asked to press a button they were holding in their hand, and that would bring up another relaxation instruction and then another emotion suggestion. And doing that, we obtained 15 1 to 5 minute periods of eyes closed spontaneous EEG from about 30 subjects. So for this analysis, we invented a new method, which we call the method of independent modulator. So after decomposing the data spatially, and selecting the brain-based independent component processes, we then jointly decomposed their log spectrograms throughout the data, trying to find modes, independent modes of spectral covariation within single component processes or across more than one. So we were looking for data-driven spectral modes. And we draw this cartoon of these modulators being in brainstem, but which is an anatomic possibility because, of course, those brainstem-centered neuromodulation systems associated with dopamine, acetylcholine, and so on, are known to modulate the EEG spectrum of the local field potentials. But here we just put them there for convenience. We did, however, find, in decomposing the data this way, three types of modes, three clusters of modes that can be only best described as high-frequency broadband. So the first type was from sources representing muscle EMG, which of course has that character. And here we show the localization to equivalent dipole localization for those EMG sources, affected sources. Here was a unique class we didn't expect. These localized to bilateral sources in or behind the two eyes. And they show a peak between 50 and 70 hertz. We looked in the literature and we found that indeed the ocular motor tremor represents movement of the two eyes, which is synchronized across the two eyes between 50 and 70 hertz, with an extent that can be as small as 1 angstrom, according to the literature. The EEG signature of this in eyes closed individual was found for several to many of these subjects by ICA decomposition and then spectral mode decomposition using the method of independent modulators. And then we found a class of high-frequency broadband modulators that pertain to brain-localized independent component processes. These are the effective sources of the brain EEG found by the AMICA decomposition. This was also unique, and it clearly wasn't limited to any one normally defined frequency band. The frequency limits of these were between 30 and 200 hertz. Although in one subject we saw that it was above 256 hertz, the limit. When we looked at the average power in those frequency bands, in those characteristic frequency bands for individual subjects, in the 15 different emotion periods, and use multidimensional scaling to project that 15 dimensions down to 2 dimensions, and then labeled the points with the emotion labels. We were very surprised to see that it was quite clear there are groups of emotion on the left, there is negative emotions on the right, there's positive emotions. To quantify that, we gave these emotion names to a group of subjects on the web and asked them to rate how positive or negative they were. And then we found that if you project in this 2-dimensional space down to this one single dimension, then the fit of the character of the EEG dynamics in the high frequency broadband modes correlated 0.96 with the ratings of these emotion words by a different group of subjects. So now we could look at where in particular, or where most strongly, did this high frequency excitement translate into positive feeling. And the answer was 2 positions in temporal lobe. Of course, we went to the imaging literature to find predecessors in that, and we couldn't. The literature at that point for emotional experience was not very strong. However, since then, we've found a number of results. This one was listening to good, bad, and neutral music. This is a bold experiment. And this one was listening to actors recite the same lines with positive or negative feeling, or neutral feeling. Both studies found these regions in temporal lobe to be those in which either bold signal on the left or high frequency EEG was proportional to varied monotonically with the degree of positive feeling, or positive to negative feeling. So you could read the valence of the emotions out from these areas. And this was a result found reported first from EEG. Now, what about imaging human agency, the willing aspect of consciousness, human actions, and also social interactions? Well, here I proposed a dozen years or so ago the term mobile brain-body imaging for a paradigm in which the experimenter records not only EEG or MEG or some other brain imaging modality, but also a richer record of behavior and motivated behavior. So if you're interested in the relationship between brain experience and brain dynamics, experience, and behavior, well, what does the brain do? Well, from one point of view, if you count molecular synaptic events per second, in your brain or mine, there's about 1 quadrillion per second, it's estimated. So that's what the brain is doing. Well, how does that translate into our coherent behavior at a dimensionality that is and a rate that is, vastly lower than that? Well, there are these emergent spatial temporal phenomena. Spikes, even from that scale, would be an emergent phenomenon. Local field potentials, effective source processes. These are macro dynamics that emerge from the nonlinear dynamics of the interactions between cellular units and supercellular units. You can now record EEG, fMRI, MEG with a resolution of something like at least 1 million bits per second, easily, or more. Yet the tradition in psychophysiology was to use as a measure of behavior a task in which the subject pressed a reaction switch, maybe on the order of 1 per second. So if you're interested in the relationship between brain and behavior, and you have such a vast mismatch between the amount of brain data you're recording and the amount of behavioral data you're recording, of course you can't get very far. And so what people would do to address this would be to average everything down to something like an ERP, pick a peak moment, pick a channel, compare the height of that ERP and that channel at that point in two different sets of responses, two different situations, and then write a grant to write another experiment. But of course, this is extremely inefficient. From an engineering point of view or scientific point of view, of course, what you should do is record much more adequate measure of behavior and then give yourself a good math problem. So I thought this was an important enough concept, and our results showed that EEG could be used to record, could do high-resolution source imaging even in subjects that were moving naturally or making even in natural 3D environments. And so I ventured the acronym MOBI, which I defined as recording what the brain does via EEG, MEG, or other brain imaging modality, what the brain experiences, not only a record of the events produced by the experiment control program, but even recordings in more natural situations of what the subject hears, of what the subject sees, and how they move, because that is what the brain organizes. What the brain organizes is our behavior. So by recording their eye movements, by recording their body movements, and their other psychophysiological measures, one now has a complete set of data with which one can look at how the brain supports natural cognition. Natural cognition is our ability to live as an agent, as an embodied agent in the world, and accomplish our goals and avoid dangers. So we did get funding to build the first MOBI lab, and of course we needed software. And this on the left is a picture of Christian Kota, who contributed some wonderful coding. The lab streaming layer is now used in many places to connect devices of different types and record synchronously from them. His extensible data format, XDF, is used by the lab streaming layer, or LSL, recording. In the lower right, Alejandro Ojeda built MobiLab, a MATLAB multimodal data browser and pre-processing application. And I don't have time to describe now, but the HED event descriptors system is now ready to describe the detailed natures of events in time series data, proposed originally by Nima Bigdali Shamlo there in the background. So what can you use the MOBI paradigm for? Well, if you combine body motion capture, video, and high-density EEG, one can, for instance, study complex motor behavior of a subject juggling some lit balls. On her arms, you see some emitters in a phase-space motion capture suit. And on her head, high-density EEG. So Hiroyuki Kambara from Tokyo Tech ran these experiments in our lab, and here he shows a quasi-Granger causal estimate of information flow between independent component processes, shown here in their dipolar location, during the three-ball juggling cycle for this subject across quite a few minutes of successful juggles. I don't have time now to interpret these results, but I think they're emblematic of what we can look to achieve in understanding how greater variety of natural cognition is supported by cortical electrical dynamics that we can see and well resolved from the scalp using these methods. Here is Grace Leslie, who was in the music technology department doing her PhD and wanted to do a cognitive experiment. So we asked the question, how can we measure listener feeling engagement in music? Are you just, hearing it, or are you really into it? And there's this...
I wish I could make you hear.
Now her friend is deaf.
Not like that. Tell me you really hear.
She wants to convey the feeling of the music. And he begins to get it. There we are, okay.
I wish I could make you hear. Not like...
I don't know why I can't stop it.
Make you hear.
So
Not like that. Tell me you really hear.
I will keep going. In the lower left, you see a screen on which the position of the subject's finger is transduced into a position of a white moving ball. We asked the subject to imagine that their friend was viewing this screen somewhere, and he was listening to music and trying to make gestures, conducting gestures that would, like this woman did in the film, would give the feeling of the music. Was it boom, 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 or was it boom, 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 etc.? And on the right, we have a lab programmer trying to do this. So there's a lot to be said, but in brief, we found 22 here clusters of brain sources, of these brain affective sources. And we looked at which ones showed differences in the two conditions. The two conditions were either you were doing this, imagining communicating emotionally to your friend watching, but not listening, or you were doing the same thing, but you also had to do a difficult mental arithmetic problem at the same time, which of course would make you less engaged in the primary task. So we found in which of these source clusters was there a difference between the more engaged and the less engaged condition. And we found there was really only one. It was in the region of the right temporal parietal junction. And you see here there was more there are theta bursts at the beginning and the end of the dominant direction swing in the more engaged condition. So we looked again in the fMRI literature and in the neuroscience literature, and we find that the temporal parietal junction in area in general controls representations of the self or another individual across processes like mentalizing, empathy, agency, discrimination, visual perspective taking, imitation, and so on. And in particular, the right temporal parietal junction is a key cortical structure for both motor control and emotion control, particularly hand motor control. And in autistics, the morphometric volume of the right temporal parietal junction predicts the subject's level of emotional awareness of others. So if we'd been so knowledgeable, we could have predicted that this task, which has to do with the imagined emotional communication with others, would be sensitive to in the right temporal parietal junction. In any case, it turned out. Another example, social interactions. This is the laboratory of Gideon Dieck at UCSD, and there's Mom on the right, and 3-year-old on the left, and they're playing a game. On the screen pops up bubbles, and they try to pop them. This is the actual recording. And you see there's motion capture, there's video, and there's high density EEG. So from this EEG of the child moving around, what can you gather? Well, this was a result by Yu Liao, postdoc who was working on the data. And as an example, this was a mu component, a hand region mu component. We can tell by the scalp map and by the characteristic mu peak, which in toddlers turns out to be 7 or 8 hertz instead of, in adults, around 12 hertz. And here we show that not only is the mu rhythm blocked, as in adults, when the subject moves, but in the child, the mu rhythm was blocked, that blue line there. The mu rhythm was blocked when he or she watched the mother reach and pop the bubble. This is, of course, highly reminiscent of what was found in monkeys with this, and the single neuron level, the so-called mirror neurons. This was not mirror neurons. This was an effective source area of cortex that showed this behavior that not only in the overt movement case for the child, but when the child watched the mother make a similar movement with the similar goal and results, did you have the same phenomenon. So I hope that this has convinced you that there's interest in these methods, and I think we're at the beginning of a fine new era of scientific exploration, functional EEG, high definition imaging, brain-computer interfaces, MOBI paradigms of all sorts, neurofeedback, brain stimulation, concurrent EEG and MEG, etc., etc. And I again thank the organizers, and we'll be happy to join the discussion.